Welcome. Today, we're diving into the world of subvisible particle analysis in parenteral products. I'm Dr. S.K. Singh, and together we'll explore why detecting these tiny particles matters so much for safety and quality in injectable medicines. Let's get started. Let's start by exploring what subvisible particles really are. These are tiny particles found in injectable products, too small to see with the naked eye, but detectable with specialized instruments. They fall into three categories: intrinsic particles, which come from the product itself, extrinsic particles, which are foreign contaminants, and inherent particles, originating from the formulation components. These particles typically range in size from 1 to 100 microns. Understanding them is crucial for patient safety, product quality, regulatory compliance, and ensuring sterility. When we talk about global regulatory expectations, we're looking at a complex landscape of standards and guidelines. USB Chapters 788 and 789 focus on particulate matter in injections and ophthalmic solutions, respectively. The European Pharmacopoeia addresses subvisible particles with EP 2919. Japan has its own standards in JP 6.07. Then there's ICHQ 6A, which sets specifications and acceptance criteria. The FDA emphasizes inspectional focus on particulate contamination control, while the EMA's Annex 1 outlines a comprehensive contamination control strategy. Together, these shape how we manage particulate matter worldwide. Let's talk about the health risks linked to subvisible particles in medicines. These tiny particles can cause serious problems, like pulmonary embolism, phlebitis, inflammation, immune reactions, and even organ damage. Certain products carry higher risks, especially intravenous infusions, biologics, protein therapeutics, vaccines, and pediatric injectables. For example, protein aggregates larger than 10 microns in monoclonal antibody treatments can trigger unwanted immune responses. Understanding these risks helps us design safer therapies and protect patients better. Let's take a closer look at where subvisible particles come from. They can originate from the environment, things like fibers, dust, or contamination in cleanrooms. Packaging is another source, with fragments from rubber stoppers, glass particles, or materials used to seal containers. Then there are product-related causes, such as protein clumps, active ingredient precipitation, or excipient incompatibility. Finally, process-related sources include filter shedding, silicone oil droplets, and even glass delamination. Understanding these helps us control and reduce particle contamination effectively. Let's look at the main analytical techniques we use to study subvisible particles. First up is light obscuration, the preferred method according to USB standards. It works by detecting particles as they block light passing through a sample, covering sizes from 2 to 100 microns. When light obscuration isn't suitable, we turn to microscopic particle counting. For protein aggregates, flow imaging microscopy gives us detailed insights. And for nanoparticles, dynamic light scattering is the go-to method. Each technique has its strengths, depending on what we need to analyze. Let's talk about the light obscuration principle and how it works. A sample passes through a sensor, where a laser beam shines on it. When a particle blocks the light, the detector notices a drop in the signal. From this, it calculates both the size and the number of particles. This method is rapid, automated, and widely accepted in official guidelines. But it does have limits. It can't tell what type of particle it detects, and it's sensitive to bubbles, which can affect accuracy. Let's take a closer look at the particle size limits set by the USP standards. For small volume injections, those up to 100 milliliters, the maximum allowed particles larger than 10 micrometers is 25 per milliliter, and only 3 per milliliter for particles over 25 micrometers. When it comes to large volume parenterals, those above 100 milliliters, the limits are expressed per container: 6,000 particles over 10 micrometers and 600 over 25 micrometers. These criteria help ensure safety and quality in injectable products. Let's walk through a calculation example for a large volume parenteral. This kind of preparation involves precise measurements and careful calculations to ensure the right dosage and concentration. We'll break down the steps so you can see how to handle the numbers confidently. Understanding this process is key to safe and effective medication delivery in clinical settings. Let's walk through a calculation example for small volume injections. We've got test results showing an average of 4,500 particles per container that are 10 microns or larger, and 250 particles per container that are 25 microns or larger. The USB limits allow up to 6,600 particles respectively. Since both our counts are below these limits, we can confidently say the product complies with USB Chapter 788. It's a clear pass. When working with protein-based injectable products, there are some special challenges we need to keep in mind. Protein aggregates, silicone oil droplets, and air bubbles can all show up in these formulations. These subvisible particles aren't just a nuisance. They can actually increase the risk of immune reactions. To tackle this, we rely on advanced techniques like flow imaging microscopy, microflow imaging, resonant mass measurement, and dynamic light scattering. For example, monoclonal antibody formulations often form aggregates during freeze-thaw cycles, which we need to detect and control carefully. When we face out-of-specification results that go beyond our set limits, we follow a clear investigation strategy. First, we review the analytical method to ensure it's working as intended. Then, we check the instrument qualification, followed by a close look at how samples were handled. We also examine environmental conditions, batch manufacturing processes, and the container closure system. Common root causes include filter integrity failure, glass delamination, or product precipitation, each one a clue to what might have gone wrong. Now, let's focus on CAPA, corrective and preventive actions. This is where we optimize processes, improve filtration, and qualify suppliers to keep quality on track. We monitor particle counts closely, using risk levels to guide us. Low risk below 50% of the limit, medium between 50 and 80, and high risk above 80%. We follow quality risk management principles, tracking trends across batches, manufacturing lines, and product stability. This approach helps us rank risks clearly and act before issues escalate. When inspectors come through, they focus on a few key areas. First, they look closely at documentation, things like validation reports, method suitability studies, and trending data. Then, they turn to manufacturing controls, checking filtration systems, cleanroom environments, and container closure systems. Data integrity is another big one. Audit trails, electronic records, and investigations into out-of-spec results all get scrutiny. Finally, they expect a solid contamination control strategy, as outlined in Annex 1. This comprehensive approach keeps everything safe and compliant. To wrap up, we've seen that controlling subvisible particles is absolutely critical for the safety of injectable products. Various global standards, like USB, the European Pharmacopoeia, and others, set clear acceptance criteria we need to meet. Light obscuration remains the go-to method for measuring particles, but biologics demand more advanced techniques. Staying on top of trends, assessing risks, and implementing contamination controls aren't just best practices, they're essential for regulatory compliance. Ultimately, effective particle control protects patients and keeps regulators satisfied. Thanks for joining me today. If you need any more support or have questions, please don't hesitate to reach out. I really appreciate your time and attention. Thank you.